Assalamualaikum and hi. Today I would like to present about my review thesis entitled as the effect of antibacterial agents in onion extract Allium cepa against aquatic bacterial pathogens. My name is Masliana Binti Zakaria, S52398. Dr. Sharifah Noor Emilia is my supervisor. In this presentation, it consists four different parts, which is introduction, methodology, result, and conclusion. Firstly, in introduction, so as we know, the aquaculture is a sector that economically increase as food contribution for human consumption and make the market demand of aquaculture product is increasing day by day, and there are many and specific plans are created to improve the production, which causing an economic loss due to spread of the disease such as bacterial infections. And due to this problem, the antibiotic were used by hatcheries to reduce the mortality, but forbidden recently due to chemical residue, especially in aquatic environment. Onion, traditionally known for their secondary metabolites that could help to treat the disease. Onion is mentioned to consist of three components that could help to treat the disease, especially in combating with bacteria. First of all, it consists non-structural and soluble carbohydrate, which is fructooligosaccharides. It's identified to have prebiotic effect by enhancing fish bifidobacteria that naturally occur in fish intestinal condition. Next, it consists phenolic compound known for their major group, which is flavonoids. Is a substance that naturally occur in plants, which is assist as antioxidant that able to protect the body from some disease. Then it consists of sulfur compound, which is known for their thiosulfonate that produce an odor and taste. The objective of this study is to review the effect of onion extract Allium cepa against aquatic bacteria pathogens. The problem are state that bacteria disease could affect the culture population and then lead to the mortalities. Antibiotics are used to overcome this issue, but is not recommended as it consists of chemical residue. In methodology, I use Prisma guideline that start from searching, screening, filter, organize and report. In searching process, 5 main websites are used to collect the data, and this is my keyword that I use. In the screening process, I screen all the title, abstract and result that should be fit with inclusion criteria. Only paper in English and Malay are accepted in this review. For the filter, the journal are manually selected by reading the journal one by one and then carefully observed to avoid misinterpreted. Lastly, the data were organized in the summary based on the result and method that used, and the data were listed in the table. 81 literature studies that we found, only 8 of them are identified to relevant with inclusion criteria. Out of 8 papers, 3 of them were studied in vivo while the other 5 were studied in vitro. So for the result, for antibacterial activity, it seems that the agar-well diffusion is the most significant method that used. A study conducted by Hanan et al. Which using reflux extraction method, it was revealed that all 33 isolates bacteria are having sensitivity effect at 12.5% of concentration. In the study, agar dilution are used to assess the MIC, and the MIC are start to appear at 90% of dilution. Next, Bello et al. Using blended method to produce an ethanolic extract, and the result showed that the Bacillus subtilis are the most sensitive compared to the least E. coli. Not only that, Bello et al. Also proves that the onion consists antifungal properties and it have the highest effect after against Aspergillus niger, which at 16.5 mm. Bello et al. Also discovered flavonoids, tannins, aconoids by phytochemical screening, which known for their function to prevention of infectious disease. This is the comparison that I have made through this study, which is a study by Itawel and Najiha et al. This study are using the same extract with methanol and water. Etawel observed that both of them have an effect, and the result were estimated at the highest effect, which goes to methylolic extract that perform it at 1000 microgram per milliliter. Meanwhile, the study conducted by Najiha et al. Performed clear zone for all types of bacteria. Takare 2004 identified that maybe this is due to specific dose that used, method of extraction, method of application, and the strain of the plants. However, the study conducted by Emilia et al. 2016, which using a sterile pastel and mortar to produce aqueous extract, did not affect the bacteria as the positive control were have the highest effect compared to the aqueous onion extract. Amre Waho et al. State that no significant difference between these studies.
For oral study which is in vivo, the feed method are the most significant to use as the onion consists sulfur compound that will attract the omnivorous and herbivorous fish. First, the study conducted by Emilia, the fish are injected with bacteria and left in the bath. The results showed that out of 30 fishes, only 8 of them were not survived. Then, the study by Epinzama et al., who test the feeding trial using fresh onion. The fish at first are injected with the bacteria and then fed in 12 weeks, and the results showed that the mortality rate of treated fish are lower than untreated fish. Not only that, Episama et al. Also revealed that the weight of the onion-fed fish are gained after the feeding trial. Then, Chu and Lee study about the onion powder formulated with fish meal into olive flounder feeding trial. The fish are injected with bacteria after 8 weeks of feeding, and the outcome show that there are no significant difference between the mortality rate, as all of the diet consists 94%. Cho and Lee state that the lysosome activity in the fish will improve as it was important index of innate immunity in the fish. Lysosome are identified to have latent activity that known to be their effectiveness against gram-negative and gram-positive bacteria.
As a conclusion, it can be concluded that the agar-agar diffusion is the best method that known to be effective against pathogenic bacteria in vitro study. But orally, onion in the feed are seems to be effective against pathogenic bacteria, especially in Itada and V. havii. Onion also provide medicinal properties such as antibacterial, antifungal, antiviral, antimutagenic, antioxidant, and so on. So in this review, which provide the previous data, which could help for future study to create fish immune stimulant against various aquatic pathogenic bacteria.
So this is the references that I use.
That's all from me. Kindly inform me if you have any question. Thank you.